Thanks again to coming to Unveiling the Power of Bioengineering by Allozymes. We have people from all over the world and all different sectors and industries, so it's really exciting to do this. I'm going to hand it over to Akbar Vahidi, co-founder and CTO of Allozymes.
Hello and good morning and good evening, everyone, and welcome to our webinar on the unveiling of power of the bioengineering. Bioengineering is a fascinating field that combines biology and engineering to create innovative solutions to global challenges. We believe it's a discipline that is transforming industries from healthcare to energy and agriculture, and Allozymes is very much eager to play a pivotal role. Today, we are going to delve into the exciting world of bioengineering, focusing on key areas like enzymes, microfluidics, and precision fermentation, and explain how Allozymes builds and uses these technologies. As many of you may know, enzymes are the workhorse of life. They are proteins that act as catalysts, speeding up chemical reactions without being consumed in the process. Imagine enzymes as tiny machines within our cells, evolved to carry out specific tasks with incredible precision and efficiency. Just to conclude, nature has shown us how to sustainably produce our everyday needs. In fact, living cells are naturally designed to build enzymes and actually depend on these catalysts for survival. When we naturally produce enzymes to perform chemistry, we are harnessing millions of years of evolution to make materials that we use every day. But first, I would love to introduce Allozymes to you. At Allozymes, we in fact started building our technology way before the incorporation of the company. But fast forward in 2020, after early stage of the technology development in university, Allozymes established and that microfluidic-based screening technology of it was patented. We moved from pre-seed of a small team to the subsequent year to raise our first large fund of seed funding, and we established our lab and 10 people to focus on our commercialization of our technology. Following year, we managed to deliver our first commercial service project to our customer and started another two, and we expanded the team as we grew. Major milestone was achieved 2023 for Allozymes, which we raised our Series A funding and started 3 new biosolutions for our own future projects and products, as well as secured 3 new services. And we expanded the team to 30 people, and which majority are scientists. This year, we have been focusing on automating our post-sorting microfluidic systems and launching our biosolutions. And we also expanded to Europe, which as we go, we will have more and more news from that operation as well in future. We believe enzymes are catalysts that we engineer for better sustainable products and for a better world. Allozymes from inception, it has been pioneering this field. We raised our Series A to build a faster enzyme development to serve more industries. Traditionally, this was heavily on pharma-oriented projects. Allozymes tried to expand this field with its own technology. And after new funds came in and more projects we started, we are focusing more data-driven approaches with the empirical data we are collecting day-to-day from the lab. We believe this will allow us to not just be smart, but with the proven data set, use that for our benefit and better enzyme engineering solutions that we will provide in future. Allozymes believes enzymes need to be engineered to boost manufacturing, and there are many examples of that. In fact, when you look at some industrial examples that are out there or coming up, oil spill cleanup that are used with alkanes, oxidases are famous, but much more interesting application is coming from plastic-eating enzymes, technically called PTAs. Some companies are pioneering that. Typically, this enzyme was found from landfill, a process that will take hundreds of years in nature, can be accelerated with this enzyme to months or years in nature, but We believe with engineered enzyme, it can be done better. For this example, now the engineered version of that, it can do from hours to days maximum, the same process. The fantastic part about engineered enzyme is it can help you to get better selectivity, efficiency, or sustainability of the process with the same resources that will you utilize in the future. However, the conventional enzyme engineering has several bottlenecks. There is a relatively high failure rate. This is not a binary complete failure, but it means within timeline and the budget of the project, many projects cannot get to their targets. It's a slow process, often takes more than a year and more than 10 rounds of screening to achieve targeted improvements of the original enzyme. And it's quite expensive process. More than $1 million is often the cost of the project in the industry, as well as the scalability of it is in question because it gives few hundred max data points per day to learn from the process. And this is, compared to the scale of problem, is very small. Okay, now I will move to next slides and how Allozyme platform builds better enzymes, and my colleague Paddy will continue.
I'm Pradeep Nair. I'm our lead protein engineer. I've been at Allozymes since 2023. Before this, I was at Zymergen, an enzymology group there, where we worked on using enzymes to debottleneck biosynthetic cascades. And before that, I was at Novozymes. That was actually where I got my introduction to enzyme engineering. There I was usually using enzymes to degrade target chemicals. So through it all, I've gotten to see how scientists use enzymes to convert what was previously impossible chemistry into routine practice. So where we usually enter the conversation is when someone comes to us and they have a chemical they want to produce. But to do so with man-made chemicals and materials would be too complicated or too dirty or too costly. And so they turn to enzymes. And enzymes for these sorts of problems are often a great solution because these are the materials that nature has evolved to control chemistry in living organisms. So there are several attributes that really make them tailor-made for this sort of problem. They can be very efficient. We'll talk about each of these in detail. They can be highly specific. They're multifaceted, and they're genetically encoded. So first, let's talk about efficiency. As Akbar alluded to, there are enzymes in the world that catalyze reactions that by themselves would spontaneously happen over the course of hundreds of years, but they can be catalyzed to produce the same amount of product in fractions of a second. So this makes enzymes incredibly useful if you want to get more product in a reasonable amount of time from your process. They're also catalysts, and that means by definition, they're neither produced nor consumed by the processes that we use them for. This means that a relatively small amount of an enzyme can be used to make a very large quantity of product, and it isn't without needing to be replenished. Enzymes are also highly specific. So they have an active site. This is where the chemistry happens. So this will be something that's specific for the molecule that the enzyme is meant to act upon. That's called its substrate. So here the active site of the enzyme will only fit a specific molecule, that substrate. And once that substrate lands in the active site and forms this enzyme substrate complex in the center, only a specific type of chemistry will happen. So there will be a certain chemical bond that's made or broken at the active site, and then those products will be released. However, an enzyme is more than just that active site. So they can do more than one thing at a time. Here, we've kind of schematically drawn an example of a cellulase. This is a very well-studied enzyme. Cellulase has that same sort of active site. Here it's called the catalytic domain, CD, but it also has this carbohydrate binding motif. So the catalytic domain is specific to sugar subunits. What the cellulase does is it sits on a cellulose fiber, which is the substrate, and it chews up that cellulose and releases sugar subunits into solution. The catalytic domain itself would successfully cleave and release one or two or more subunits, but it's linked to this carbohydrate binding module. The carbohydrate binding module sticks to that fiber and basically feeds more sugar subunits into the catalytic domain. This allows the cellulase to be highly processive, meaning that it can produce many, many molecules of sugar from one attachment event to that cellulose fiber. So working together as an integrated machine, this single enzyme will specifically cleave and produce one type of product many, many times without needing to reattach to that cellulose fiber. And the final attribute that we take advantage of here is that all of these enzymes are genetically encoded. That means everything we've talked about so far, that catalytic efficiency, the specificity, and the multifaceted nature, so the different domains of an enzyme, they're all controlled by the A's, T's, C's, and G's in the DNA sequence of the enzyme. So that DNA encodes different amino acids. There are 20 naturally occurring amino acids. So if you just wanted to think about how many combinations of a single amino acid protein there were, you would have 20. If you thought about how many pairs, it's 20 times 20 squared, so that would be 400. These numbers get very large very fast, right? So a typical protein might be 300 amino acids long. If you wanted to think about the number of combinations of amino acids in that 300 amino acid protein, it would be more than the number of atoms in the universe. So we can't experimentally test every single one of these possibilities to find the best one. And this is really where Allozymes can accelerate this whole process. So we use directed evolution. So we start by designing libraries for a given target enzyme. And those designs can be from a number of different concepts. One that's design free is called a scanning library. So that's where we take every single substitution possible, and we do it one at a time across the amino acid, across the protein sequence. We also can look at sites that might be near the active site, or we can look at amino acid residues that are evolutionarily conserved in related enzymes. Or we can use more AI-driven approaches that'll actually be the subject of a whole different webinar. But we can make libraries along each of these different types of concepts, might have 5 different flavors of enzyme libraries. We can then order the DNA to experimentally build those in the lab. And then we construct those. Maybe we construct 5 different enzymes in a week, enzyme libraries in a week, each containing maybe something on the order of 1 million different enzyme variants. And then we can use our microfluidics platform, which Patrick will tell you about in just a minute, to test each one of those libraries. And this is the key. So maybe some of those libraries are duds. Maybe we didn't have any good enzymes in that pool. AI is getting better every day, but there's still a lot we can't predict. But the key is that we can build multiple enzyme libraries, and we can screen them very quickly and deeply, and we can see which concepts. Was it maybe that active site concept, or maybe it was the evolutionarily conserved concept, but which one really lit the fire under our enzymes? And then we can select the best enzymes, and then we can validate those enzymes using standard analytical chemistry approaches like HPLC or gas chromatography. So in this manner, we can really accelerate the process of directed evolution and get to the enzyme that's going to be best for our process. So now I'll hand it over to Patrick. He's our scientist in charge of the microfluidics platform. He can tell you more about how we do that.
Thank you, Pradeep. Hi, my name is Patrick. I'm the senior scientist in charge of the microfluidic department at Allozymes. Here in Allozymes, I developed the microfluidic platform for ultra-high throughput sorting of enzymes using our proprietary microfluidic chips and technologies. Using the microfluidic technologies, we're able to think small and achieve big changes in the field of enzyme engineering. To start, I shall first briefly explain what is microfluidics. Microfluidics involve the manipulating of tiny volumes of fluids in channels with dimensions on the order of micrometer. To have a better understanding, the channel that we use for enzyme screening and sorting are about the size of a single strand of hair. The two pictures on the right shows typical examples of microfluidic chips that are commonly used. Microfluidic is widely used to develop tools for cell biology. This is because the microscales of microfluidic device is particularly adept to work with cells, and the miniaturization of the system allow us to easily implement high throughput assays. Moreover, the fluid properties at these scales helps to create stable concentration of gradients and to precisely control fluid compositions and temperature. Microfluidic technologies offers numerous advantages in bioengineering. By simply miniaturizing macroscopic system, we achieve a huge reduction in volume needed for analysis. This translates to the use of less volume of samples, chemicals, and reagents that is needed for enzyme screening and sorting. The manipulation of fluids at the microscale exploits the benefit of laminar flow, which enable precise control of liquid composition at subcellular resolutions. The high surface area to volume ratios in droplets allows rapid heat and mass transfer and make them ideal for efficient and safe chemical reactions that can be precisely controlled and monitored. So in summary, the use of microfluidic technologies at Allozymes reduces the reagent consumptions, provides a highly controlled environment which are also highly reproducible. This process can also be parallelized, automated, and portable. In Allozymes itself, we make use of mainly droplet-based microfluidics as a microreactors. In these droplets itself, we normally encapsulate E. coli or various cells as substrates together with the enzymes. These droplets are often formed at a rate of 3,000 droplets per second. This translates to about 2 million different reactions in these microreactors in a short span of 10 minutes. Pico injection is needed for almost any assay of practical importance. This is because most assays require several reagents to be added at different times. Our proprietary Pico injectors allow us to add controlled volumes of reagent using electro microfluidics at kilohertz rates. At Allozymes itself, we hope to transform enzymes engineering with our cutting-edge microfluidic technology. At Allozymes, we deliver unique enzyme sequences using our microfluidic powered technology. This is achieved through our proprietary chip designs and post-sorting automation. So with this, I will pass on to Shirley, which will explain on our sequencing analysis.
Hi, everyone. My name is Shelley Cheng. I'm a senior scientist in the protein engineering group at Allozymes. So now, carry on from Patrick's topic, I'll talk about sequencing analysis. So as Pradip said, there are so many different possibilities in a library, and we can really go in and pick up every single cells to understand the sequence. So we rely on sequencing to decipher our sorting results. So at Allozymes, we use nanopore sequencing mainly to understand our libraries. So here's a very simple illustration. You can see we have a pre-sort library, which that could be a million of different variants in there. So after microfluidic sorting, we'll enrich what we call a sorted population. We call it a post-sort pool. So in this pool, we'll have only limited number of variants that has better activity. So how do we understand this? So using sequencing, we develop in-house wet lab and dry lab workflow to understand the whole the complete gene sequence. So we can understand the exact sequence of every single gene of interest inside a pool. Using our wet labs method and dry lab method, we can decipher up to 1 million DNA molecules inside a pool. So by understanding all these information, we can understand individual sequences, and we also calculate the variant occurrence inside our library, pre-sort or post-sort. So how do we do this? Let me show you this very simple examples. So here you can see in a pre-sort library, I'd say it's a scanning library, we have a parent protein sequence here. It's a 10 amino acids illustration. Here we have example of 4 variants. We have, each of them has 1 mutation, so ACHD, 4 variants. So in here, if you look at this yellow variant, the H1, the H variant here, so let's say if this variant has high activity, we can say enrichment in our post-sort pool. So in the pre-sort pool, we'll say this variant has 25% of the population, right? So it's 1 out of 4. When we get into the post-warp, we can say it's 70% of population. It's 3 out of 4. So 75 divided 25, the enrichment factor is 3. So this is kind of how, the general idea how we calculate enrichment in our sorting. So how does it work out in the real life? So here is an example of a real data in an enzyme with 300 positions. So each of this, the columns are positions, so 300 of them. And the y-axis is substitutions. So remember, we have 20 amino acid. If we don't calculate the wild type, there are 19 possible substitutions. And each tile in this heat map indicates one unique variant. Okay, so here you can see the percent population of each variant ranging from 0.004% to 0.129%. And it's mostly homogeneously distributed. The median of these populations is about 0.2%. 0.02%, with some a bit higher. But here is our input library. You can see it's a pretty good quality. We cover the whole sequence space. So after sorting, here's our result. You can see there are a lot less variants in our sorting. That means we already weed out all the non-functional one or low activity ones. So here what we're seeing is after activity sorting, we're enriching the winners of this particular activity that we're looking for. Again, you have x-axis of different positions, y as different substitution, and the highest population percentage or highest enrichment factor calculated. In the end, we have 1 winners. And here we down select from 6,000 variants down to about 100. So we can carry out follow-up different validation tests to really understand the activity of these variants and pick the final winner. So this is based on our pool sequencing result. We can also do clonal sequencing. So we have in-house workflow to do high throughput clonal sequencing up to 8 times 96 well plates. So using this method, we can identify the sequence, the variant of each individual wells, and link it up to the functional data. So it could be plate-based method, fluorescence, absorbance, or HPLC data. We can do high throughput HPLC as well. So by combined sequencing and HPLC data, we can input all these information into our machine learning model and to help us predict next round of improvement. So next, we'll pass the stage to Balaji. So he'll talk about our precision fermentation platform.
Hello all, I'm Balaji, strain engineering scientist at Allozymes with a PhD in metabolic engineering. I am with Allozymes from 2021 and contributing to different enzyme engineering and strain engineering projects. I am here today to share more about precision fermentation done at Allozymes for the production of high-value chemicals where we apply the engineered enzymes as presented by my colleagues before. Compared to an enzymatic reaction with a single enzyme, precision fermentation is complex and involves multiple enzymes to perform a series of cascade reactions. The cascade reaction starts with a simple substrate as a sugar and flows through the synthetic pathway and gets transformed into a product of interest by a series of enzymatic reactions. This complex reaction is possible as it happens inside the microbial cells, and we call these engineered microbes as microbial cell factories. Fermentation, the traditional method of producing products like bread, wine, and beer, uses natural microbes, whereas precision fermentation leverages cutting-edge biotechnology to engineer these microbial cell factories to produce a wide range of valuable products. It can be from biofuels to pharmaceuticals all the way to food ingredients. By carefully manipulating the genetic makeup of these microbes, scientists can create strains that efficiently produce desired compounds. And some exciting applications of precision fermentation include producing sustainable alternatives to animal-derived or plant-derived products and developing new drugs and vaccines. Why do we need microbes for executing this complex reaction? As shown on the left, microbes naturally convert sugar, such as glucose, to energy-rich precursors while performing thousands of enzymatic reactions for its own metabolism. By tapping into these precursors for our target production, we are leveraging the efficiency of naturally involved pathways while using simple and sustainable starting materials. As shown on the right side, enzymes require different cofactors such as ATP, NADH, NADPH as energy sources for completing a biocatalytic reaction most of the times. And microbes generate and provide these cofactors for executing our own target reactions. And these cofactors are also continuously recycled with the metabolic pathways of the microbes. This slide shows the full overview of a precision fermentation happening in Allozymes. First, to convert the sugar feedstock all the way to pure high quality product, interdisciplinary research and development is required. Allozymes have these R&D expertise in-house. Let me explain step by step. After choosing the feedstock, strain engineering is initiated. It involves genetic engineering to add or remove enzymes or to tune expression levels of the newly added enzymes. And then enzyme engineering is required to develop a better enzyme for the process. Then comes in metabolic engineering that is done to effectively incorporate the synthetic pathway with the naturally occurring metabolism inside the microbe. During this whole process, bioinformatics provides the in silico support and biochemical characterization provides the analytical methods to understand the process. Finally, there is omics, which gives us a detailed picture in different levels such as transcriptome, proteome, and metabolome. So all these different fields come in together to develop a perfect strain. But it doesn't happen overnight. It needs to go through a few rounds of development. And the developed strain undergoes process development, which is the next stage, which the next speaker will elaborate more. Strain engineering and process development work hand in hand in Allozymes and undergoes a few cycles of DBTL, which is design, build, test, and learn to develop an efficient microbial factory. This microbial cell factory, which is validated in our lab, is then given for scale up in different levels, and finally, a pure product is commercialized. Though there are several synbio players in the market doing precision fermentation, the upper hand of Allozymes is its enzyme-targeted engineering. Let me explain how. Almost all the process, as shown on the left, almost all the process that go from idea to commercialization have to cross this steep called valley of death here. So this is where you have limited resources to develop a process that is commercially viable. To increase the commercial viability, it is crucial to reach the targets in three very important performance metrics, which is shown as here: TRI, titer, rate, and yield. By quickly achieving the targets performance metrics of TRI, you can cross the valley of death faster and significantly increase the chances of commercial success. As shown on the right, there are four promising engineering steps that have been well studied for several years to increase the TRI, which are: first is increasing the precursor and cofactor supply. Second is eliminating bottleneck enzymes or steps in the pathway. Third is improving the enzyme specificity to not produce any byproducts and also pull your product flex completely towards your product. And fourth is downregulating competing enzymes or pathways not to waste your carbon. All of these steps require some forms of enzyme discovery and enzyme engineering to bring up the efficiency. And by doing engineering, you are significantly increase the cap of maximum production in this process, thus quickly achieving the tighter rate and yield. So the bottom line here is efficient enzymes are the key to increase tighter rate and yield, and Allozymes is doing that using its enzyme targeted engineering. Here I have a case study where we have developed a precision fermentation process for sesquiterpenoid production using engineered E. coli with Allozymes expertise. So sesquiterpenoid is a bioactive chemical which is derived from FPP that have anti-inflammatory, antioxidant, and several other valuable properties. Currently, these molecules are mainly extracted from plants, which is not sustainable. So we have developed a microbial platform for producing this sesquiterpenoid in large scale. For that, we have first assembled a hybrid pathway in E. coli involving 15 to 20 enzymes. And during the process, we have discovered, assembled, and expressed several non-native enzymes into the E. coli. Then we identified the bottleneck in the pathway. We engineered the 3 key enzymes in the pathway to increase titer, which is one of the key metrics. Then we developed an optimized bioprocess to increase rate, which is another very important metric. Further, we balance the pathway flux because the cell needs to grow while producing this sesquiterpenoid. So you have to balance the pathway flux. You don't want to completely kill the cell, and you don't want the cells to just grow without doing any of your production. So you have to balance the pathway flux using metabolic engineering. Thereby, we have increased the yield of this process, which is the third metric. Finally, we have a downstream process developed in-house by the next speaker, Sanjay. And with that, we have isolated the product with high purity and made this process ready for commercialization. Now, I pass on to our next presenter, Sanjay, who will expand more on our bioprocess capabilities.
Hi, hello everyone. My name is Sanjay D'Souza and I am a senior DSP scientist at Allozymes, where I have been working since February 2024. Prior to this, I spent several years in Indian biotech companies, specializing in the purification domain. My work focused on developing cost-effective, scalable processes for enzymes, proteins, peptides, and probiotics. I have been deeply involved in various aspects of their research and development process, including evaluating proof of concept studies at the inception stage, characterization, optimizing each step, and eventually facilitating technology transfer to the production scale. I am excited to be part of the Allozymes team and contribute to realizing the vision of our founders, Dr. Payman and Dr. Akbar. The upstream process. The first process in the bioprocess is upstream, where in a known strain is grown in a fermenter. At Allozymes, we have 1.5 liter to 14 liter fermenters, wherein we optimize and scale up the process. At 1.5 liter, it is a geometric volume, and we work with 1 liter there. And in 14 liters, we have a 10 liter volume. And these are all fully automatic. We can monitor and control the fermenters in a real time. These fermenters also can be run in a batch mode, fed batch mode, and continuous mode. The 1 liter fermenters will help rapid testing of media, pH, and temperatures. At higher scale, that is 14 liter, we call it as a pre-pilot scale, where we optimize all the parameters before we move on to a bigger one, that is 100 liter. This is a 100 liter fermenter where we geometric volume is 150 liter, but working volume, what we operate is about 100 liter. And this is an industrial scale down model where complete steam sterilization can be handled automatically, and also media can be sterilized, and it completely incorporates CIP and SIP and the pressure hold test. It automatically adjusts the fermentation conditions such as feed and pH control, and which has a SCADA system in it. Moving on to the downstream side. So the moment the upstream process goes on, parallelly also we start understanding the steps that required for carrying out the downstream process. The general downstream processes are cell lysis, cell separation, product concentration, chromatography, drying, and packaging. The very steps I would say is the cell lysis, but it also depends on the product expression. If the product is produced intracellular, then only this step can be implemented. If not, then straight away we move on to a cell separation. So for cell lysis, we have three methods mainly categorized as physical, chemical, and biological. So in physical method, we use homogenizers, sonicators, and manual cell grinding also used. Among these, homogenizers are considered to be more reliable and they are scalable ones. In chemical method, surfactants, alkali, acids, and solvents are also tested. In biological approach, we use enzymes which can lyse the cells. Once the cell is being lysed, the next step is a cell separation, wherein most of the insoluble components and active cells are removed from the media. So several instruments are used in these steps. It can be tabletop centrifuges, it can be continuous centrifuges, also filtration system. Basically, the models which are of industrial use is like the filtration model and the continuous centrifuge. At Allozyme, we have a filtration system where we evaluate and optimize the steps here for the next step. The product concentration. Soon after the product is being centrifuged and the suspension and the dead cells are removed, the product remains in a diluted form. Hence, it needs to be concentrated for further process. So for this, filtration systems can be tested, and secondly, also resins can be used. At Allozyme, we have a system where it can be concentrated to certain extent and processing is done. Also, we have a couple of resins which we can test and understand the absorption and concentration part. Chromatography step. In this step, it is, I would say, it's a polishing step wherein you remove all the unwanted impurities and get the product to a desired quality. So the system has a pumping module and also the column where different resins like polar and non-polar resins are tested and the process is optimized and product is collected. The drying step. This step really depends on the format of the product. If the product requires to be in a solid format, then vacuum drying, freeze drying, or spray drying can be utilized. Vacuum drying basically used for the semi-solid products where moisture is about 50 to 60%, and freeze drying is for the products where they are very temperature sensitive. Spray drying is a very economical drying process for the food products such as milk and product and dyes. The final step I would say is a packaging and storage where we evaluate the temperatures and humidity to enhance the shelf life of the product. Also the closure container system where it is compatible to store it in a longer duration. So the next, I halt for the process, and the next slides will be biosolution license.
All right, thank you, Sanjay and the team. So I will just explain what we are doing in action. We explain what platform we have built and how we have been working on enzyme and strains for commercial users. I believe our announcement about two partnerships this year kind of abstracts how our platform works. One is with Adesio. Adesio is one of the leading companies in the animal feed, and they are one of the top companies for methionine production worldwide. We are in this partnership, we are helping them to improve their enzymes in their processes over the R&D phase projects they have. And they are building cutting-edge new technologies for more sustainable development of animal feed. In this partnership, mostly we are debottlenecking their enzyme cascades within the biocatalytical pathways or strain pathways. And the development will be commercialized by Adissio, and Allozymes heavily working on the early stage and the piloting stage of the development. On the other side, we have also ScienceCo partnership, which we believe with this partnership, we are helping decarbonize home and personal care industry. ScienceCo is one of the leading chemical companies in Europe, recently spirited from Solvay. We are working to commercialize several ingredients, especially sustainable specialty ingredients portfolio that they aim to commercialize. Allozymes' edge here is we can improve enzymes that will build much more efficient pathways with better titers and yields that can be used for commercial production of the ingredients. And again, the commercialization will be taken care of by our partner in this case. These are great examples of how innovative technologies can help the bigger players that Allozymes aims to achieve. With this, we will end our webinar that we have prepared to present, and we are more than happy to receive your Q&A and start responding on that. I think John will moderate that part, and he requested for everyone to share if they have any questions, of course, as much as time allows. Thank you, everyone.
One question we have, I think it's on precision fermentation. How do you address challenges like pathway optimization, and how do you ensure efficient production of target compounds? So maybe we can move that to Balaji, who discussed precision fermentation. We have one more question here. Can you discuss the specific methods Allozyme uses to modify enzyme properties, such as stability, specificity, and activity? And how do you balance all of these to achieve optimal performance? So I'll move that over to Pradeep.
One point I would say just at the outset, a difference between doing like plate screening versus doing sorting that we do here is that we don't necessarily need to balance them. We hope to get the best of both worlds. What I mean by that is when doing plate screening, for instance, if you want to engineer specificity, what will often be the paradigm is you will make a library of enzyme variants, and you will have substrate 1, which might be the wild type substrate, and then you'll have your desired substrate. And you will screen all of those variants across the 2 different substrates. And so however expensive and however long it took you to do that screening, it'll be ×2 or however many substrates you want. What we can do instead, for instance, with specificity, is we can design an assay that can be used for detection of both reactions happening. We can run that library through our sort. We can find variants of the enzymes that are active, because many of these mutations will just kill the enzyme. So we remove those mutations, and then we can take that pool of winners and we can screen against the second desired substrate. So in that way, we've already ensured that we have some activity by doing that first to remove the deads. And we've now taken the subset of those variants that are going to be active against the desired substrate. So that's specific for, that's to engineer specificity. For stability, what is often done is to add in some sort of either thermal shock or chemical shock. So for instance, we have a project where they want to increase stability, thermostability of an enzyme. And so there you typically, you treat the sample that contains your enzyme with whatever perturbation you're trying to make it protected against. And then you assess the activity of the enzyme. So that's all about the assay part. I will say there's a whole separate part of it, which is the data science part. How do we design libraries that will have a better potential to yield hits. I think that that's actually best for us to talk about in a future webinar. So stay tuned for that.
Okay, we actually have one last one that just came in. Let me read that up. How to screen or sort engineered enzymes for which the activity inside droplet is limited due to factors such as substrate solubility, product detection limitations.
Thank you for the good question. So for product solubility, if that's the question, of course, we do have a range of solutions that we test first to increase that solubility. And also, I would just mention that scales in the droplet is very different. You normally work with single cell and a limited number of enzymes, so the concentration should be correlating to that. So we don't necessarily need to achieve very high concentration of these substrates. But we also have some other ways of certain miscible co-solvents that we use in our droplet system, which is compatible and helps with hard to dissolve, handle the hard to dissolve substrates. So that's how we achieve them. With that method, we can achieve quite significant amounts, but these are not always needed in the screening for us. And in terms of the, pardon me, I check the second part of the question, detection limitations. Yes, if there are if there is a limitation in terms of initial activity is very low, there are ways we can amplify that. For instance, we can grow the cells in droplet and some other methods that we have established and used in our projects to deal with very low activity, or we call noisy kind of situations. And one of the cases we had barely detectable activities, and we managed to improve that with 100-folds with the methods I mentioned.
All right, so I think those are all the questions. We really appreciate everyone coming in. Like I mentioned before, people from all over the world and different industries. It's really exciting. This is unveiling the power of bioengineering at Allozymes, and in the near future, we have a few more webinars, maybe some stuff on data and machine learning and all that good stuff. But till then, stay tuned and follow us on our socials. Thank you, everyone.